Good morning, students. For Standard 12, we will continue with the lesson on sexual reproduction in flowering plants, particularly the angiosperms. Now, in the last class, I was telling you about the development of the male sporogenous tissues, or what we call the male gametophyte, or the male anther very precisely. Now, the anther lobe, if this is a cross section of an anther lobe. Now here the connection here is actually in this particular point, which is connected to the filament, and it has two lobes. This type of anther lobes having two lobes is called diathecous. Now most of the flowering plant, they have diathecous type of the Microsporangia. Now inside this there are this type of sac-like structures are there, which we call the Microsporangia. Now this is the ultimate state where almost the development has taken place. Now, when the development of the anther takes place, usually it takes place from the type of archisporial cell, and that archisporial cell is the outer epidermis, and that outer epidermis divides into three layers, and the endothecium, the middle layer, and the tapetum. So this is forming the outer layer, and the inner layer there is a primary sporogenous tissue. Now this primary sporogenous tissue is enclosed within this layer of the tapetum. The middle layer usually are ephemeral. That means ephemeral means when the anthers are mature, the middle layer usually get destroyed. Okay, but the endothecium that remains. Now this there are four spore sacs like this. And inside these spore sacs you have the microspore mother cell. Now the microspore mother cells, what we call the pollen mother cells, these are diploid cells initially, but they develop from the microspore mother cells, which are diploid, and through rapid mitotic division they form the primary or what we call the pollen mother cell. Now the pollen mother cells are diploid, but later on they undergo meiosis and develops into four haploid cells. Now that will come later on. Now you can see this type of four microsporangias are there. Inside this, the development of the pollen grains, or what we call the male gametes, take place. Later on, when the anther is matured, usually this two layer, the middle layer usually is destroyed, and this sporogenous, they become a single sac. Now when in both the sides. Now what happens is that usually they dry up. Here this cells dry up, and here there is a small pore created that is called the stomium, and through this stomium these anthers come up. Because of the drying of this wall, there is a breakage of this wall here, the mouth type of structure is formed, and that is called the stomium. So the pollen grain comes and sticks all over the surface, outer surface of the anther lobes, and from there they are carried by different types of agents, and that we call the pollination. So that's a different subject, that how they are pollinated with different types of agents, like wind can be a pollinator, insect can be pollinator, animals can be pollinator. So various other agents can be pollinators. So this is how the development of the anther takes place. Now we'll come to the formation of the microspore. I told you that the primary sporogenous tissues, which are contained in this microsporangia, they mitotically divide to form diploid, the microspore mother cell. These microspore mother cells are diploid. So there is a, pollen grains are numerous. So there must be numerous microspore mother cells. So these microspore mother cells are formed from the mitotic division of the sporogenous tissue. So this diploid microspore mother cell now undergoes meiosis. That means a reductional division to form the four haploid microspores. This means they contain half the number of the chromosome of the plant. Now these are called this isobilateral tetrad. The remains attached to form the isobilateral tetrad. So initially, if you see, the pollen grains are attached like this. Here you are seeing actually three, but there are not three, there are four. That means 1, 2, 3, and one on the reverse side. Here you can see this is they are lying on the same plane. This is a type of parietal. Here it is T-shaped. So you can see the different arrangement. Here there is a linear arrangement of this microspore tetrad. So these are called isobilateral tetrads. So these are the different types of the arrangement of this pollen tetrads. Now the study of pollen grain is known as the palynology. Now you have to know this because it comes as a small 1 mark question, it can come. So palynology is the study which deals with the pollen grains. Now when we take a structure of the pollen grain, the pollen grains are, they have varieties of shapes. Sometimes you see they are oval, sometimes they are elliptical, sometimes they are round, sometimes they have a very rough surface, sometimes smooth surface, sometimes they are dusty, sometimes they are heavier. Now usually each pollen grain which contains there are two walls, the outer wall we call the sporoderm. The sporoderm is the outer wall which is called the exine, and an inner wall which is called the intine. So the exine is actually in between the exines there are small pores which are called the germ pores. Now the quantity of the germ pore may vary. Like say for example in a dicot plant there may be 3 germ pore. In a monocot plant there is a single germ pore. So usually through this germ pore the intine wall breaks out during the germination of the pollen grain, when the pollen grain falls on the stigma or the stigma of the plant. There are 2 types of nucleus. One is called the tube nucleus and the other is the generative nucleus. The function of the tube nucleus we'll know later on, that is mainly to direct the formation of the pollen tube towards the micropyle of the ovary after the pollen pistil interaction. Whereas the generative nucleus, this divides later on to form the 2 male gamete. We'll study in the next chapter regarding the germination of the pollen grain and also the process of pollination and fertilization. So as I told you, sporoderm consists of 2 parts. One is called the intine, another is called the exine. And the exine is, the intine is thin and the exine is thicker, which consists of a substance which is called the sporopollenin. Now sporopollenin is said to be one of the most resistant substance, biological resistant substance. So it is made up of polymers of carotenoids and carotenoid esters. As I told you that if the pores are 3, as you find in the dicot plant, we call them the trichoplate. If the germ pores are 1 only, then we call monocoplate. That means in monocot plant we have the monocoplate germ pore. In a dicot plant usually the germ pores are 3 in number. Now what is a pollen kit? Sometimes these exines are very variable, they have a variety of structure, and the exine is made up of a sticky substance. Now this sticky substance which is found on the outer wall of the pollen grain is called a pollen kit. Now this pollen kit attaches or adheres to the surface of animals, or it can adhere to the surface of the insects, which helps in pollination. Now the pollen grains are sometimes very allergic to some people. Like you have heard about a grass called carrot grass or parthenium. Their pollen grains are too much, it causes allergy of the skin and can cause skin eczema on the skin. So some people are very much reactive to the pollen grains, and their body, the immune system, they react to that and they have allergy. Usually you will see people, those who are suffering from asthma and all, they have to flee certain of the areas where there is a lot of flower because of the allergen created by the pollen grain. Now pollen products are also like this sporopollenin which I told you. They are nowadays used as a nutrient. It has been seen that they contain a lot of substances which are good for athletes. So they are converted into certain types of nutrient, mixed with variety of other substances like honey and all, and usually used by athletics people, those who are related to athletics and sports. Now the viability of the pollen grain. Pollen grains are viable, like say for example, some pollen grains are viable for say 30 minutes, like rice and wheat. When they fall on the stigma, or if they are falling somewhere else, they can be alive for 30 minutes. After that they are destroyed. But there are certain pollen grains, like for example in the family of Fabaceae, in the family of Solanaceae and Rosaceae, where they can remain as such, alive for maybe for months. So the stability of the pollen grain varies. So this variability of the viability of the pollen grain has been of great importance, as because these pollen grains nowadays, what they are done, that some of the rarest plant with their pollen grains are stored in liquid nitrogen. Say they are stored at -193 degrees centigrade, and these are called the pollen banks. So pollen banks are, you can see, a type of gene banks where you can store a particular pollen grain which can be used later on for fertilization of some of the species of plant which are getting destroyed, or what we are losing those plants because they are not adaptable, the adaptability is not so good. So those plants can, the genes of those plants can be stored in this type of pollen banks, which is a type of gene bank. Thank you very much. This is what we have for today, and stay safe. If you have any question, you can put up the question, and once again, thank you.